I'm Daniel Levine, and this is the Bio Report. Transcription factors control the genetic programs that maintain cellular balance, but while they've been seen as compelling targets for aging-related diseases, they've long been considered undruggable. Junevity's Reset platform leverages large-scale human omics data and AI models to pinpoint key transcriptional drivers of disease and to design siRNA therapies that restore healthy gene expression. We spoke to John Hoekman, co-founder and CEO of Junevity, about the role transcription factors play in both healthy biology and aging-related diseases, the company's AI-driven platform for identifying dysregulated transcription factors, and its lead programs targeting type 2 diabetes and obesity. John, thanks for joining us.
Yeah, thanks for having me.
We're going to talk about transcription factors, Junevity, and its AI platform to develop siRNA therapies to address significant chronic diseases of aging. Let's start with transcription factors. What are they, and what role do they play in healthy biological functioning?
Yeah, so transcription factors are a certain type of protein in the body, and they're actually very widely expressed throughout the body and in the human genome. So about 10% of the genes that we know of are transcription factors. And so when you think about proteins, they have lots of different jobs throughout the body. Transcription factors are almost like a manager within the body. So the DNA has lots of instructions for different programs to do different things in various cell types. And the transcription factors are the ones who are getting the signals from the environment, from other tissues in the body. They're getting all these signals, and then they're going around saying which proteins should be expressed or which programs should be run in various cells. So they're super important in keeping everything in kind of a constant homeostasis, especially as you get older. They're very, very important in early development. So a lot of things we know about transcription factors are from early development. They actually guide, say, cell differentiation. To take a stem cell, and they are the ones who say, turn on these proteins, we're going to turn into a skin cell, or turn into a bone cell by activating these programs. So very, very important, very ubiquitous throughout every type of cell, and they have different functions throughout your life. But they're also highly implicated in aging and in a lot of different diseases, which is why we at Junevity are very focused on them.
What role does their dysregulation play in diseases?
Yeah, so as your body ages and you get out of that development phase, I think, especially in the age when you're a baby, and then when you're going through adolescence, your body's doing a lot of things to grow into an adult and to get your organs to the size that they should be and to get everything functioning properly. Transcription factors are highly involved in those steps. They act through, and they also affect things like hormonal transmitting, which we think about in those stages of life. But once you're an adult, you pretty much want to be in a nice homeostasis. The ideal will be if you're eating well and exercising and living a nice lifestyle with not too much stress, then the transcription factors will go and just set out on their task of managing the body as like If you think about managers in a company, that it would be a company that's just trying to make the same, 10% profit margin every year, and nothing's going to change, and we'll just keep moving along like this, and it'll work nicely. The problem comes in when you have either driven by external stimuli, environmental stimuli, or aging, where you have the transcription factors. All of a sudden, they're not working as efficiently as they should, or some of them are upregulated or downregulated in ways that they shouldn't be in various tissues. And so you have transcriptional, what we'll call transcriptional dysregulation or dysfunction, where even a transcription factor being overexpressed by, let's say, 20%, 30%. So it's not 100-fold higher. It's not there totally out of control in an obvious protein that's just like expressed to a crazy degree, but it's just turned up. Like it's a knob that's turned up a little too high. That can lead to a ton of downstream consequences, such as many proteins and programs within the cell becoming out of sync. And then this leads to a pretty bad feedback loop, which can then exacerbate the disease. Some of the very obvious ones. Oh, I should also mention this. This can also happen with aging. So as you age, there's things that happen with transcription factors where just naturally they drift out of their optimal state, and that can also lead to things with aging that all of a sudden start breaking down, and these changes build on top of each other. So some of the real obvious examples with transcriptional dysregulation driving disease. One is cancer. That's probably where transcription factors are the most well studied because a lot of money has gone into cancer research. But there you definitely have transcription factors that usually tell the cell to stop multiplying. Those transcription factors get downregulated, and then you have other ones which protect against it and kind of drive cell death, and those become downregulated, and then you have cancer cells where all the checks and balances are gone, and then they just start growing and transforming into cancer cells.
If you're targeting a specific disease, let's say type 2 diabetes as an example, is targeting the transcription factor a viable way to treat the condition across the board, or do you somehow need to identify subpopulation of patients where the transcription factors are dysregulated?
Yeah. So type 2 diabetes is a great example of a disease that is, age is a big risk factor involved in type 2 diabetes, but it's also, very widespread throughout the population. So to that point, there are multiple drivers of the disease, and patients tend to have, the different profiles depending on where they are in the stage of the disease and also their internal genetics. Um, One of the things that drew us to type 2 diabetes at Junevity as a disease that we wanted to look at from a transcriptional problem is that type 2 diabetes, there's a lot of things going wrong in the body. It's highly, again, one, driven by aging, but also external stimuli. It's something where it builds upon itself because of these stress signals that just keep occurring and keep building into the disease. I think the way we looked at it from the get-go with type 2 diabetes or a different disease, instead of kind of looking at existing biomarkers that would say this person is this type of type 2 diabetic, or this person is this type 2 diabetic, we started looking at uh, very basic, large-scale transcriptional data in basically as many patient types and data sets as we could get our hands on. And I think that's the approach that Junevity is taking, which is very unique. We've built this, what we call a reset platform, which starts at the base data. And instead of giving instructions to our algorithm to say uh, patients fit within this class or this class, and giving rules to figure out which pharmacological pathways are out of order or could be targeted. We're taking it more like a large language model or machine learning approach and saying, here's the data, here's all these different patients. We're going to look at how transcription is out of whack in these patients and from a large scale networking standpoint. And then we're going to see how that replicates across patients and across data sets to try to find essentially what are the pathways that are driving the majority of cases where you see homogeneity in the transcriptional changes that we're doing. And I think that's a fairly unique approach. I think one of the reasons it works in type 2 diabetes is that even though it's a very large patient population, about 80 to 90% of type 2 diabetes patients, the disease is driven by the combination of um, eating, food intake, and exercise, the lack thereof. And so you do have, even though it's a wide, diverse patient population, the drivers of the disease are fairly homogeneous, and you could target. And our theory is you can target a large swath of the population with this type of approach.
Transcription factors have traditionally been very difficult to target for a variety of reasons. What are some of those challenges?
Yeah, so it's certainly not a lack of importance in the body. I think it's been recognized that transcription factors do are involved in a lot of different diseases. And again, cancer is probably the one that was investigated first and most, just because of the sheer amount of pharmacological discovery work in cancer. But one of the reasons why these haven't been looked at as drug targets, part of it is just historically the way the pharmaceutical industry has looked at and kind of refined potential targets is looking at it through the lens of small molecule drug development. And small molecule drug development is small chemicals that usually fit into a pocket of a protein. So they're very stable molecules. They're usually orally bioavailable. So if you think about, you can put them in pill form, go into the stomach, they get broken apart, and then get absorbed into the intestine, get into circulation, and then pretty much target many cell types throughout the body. If you can't find a small molecule that can go through that process of being an oral small molecule, historically those have just been ignored and pushed aside and said, well, if we can't make it into a pill, probably not going to make it. And transcription factors are one of the worst types of targets for small molecules to bind to because of the shape. They don't have little pockets to fit into where they can bind and alter the activity of that protein. They tend to be dynamic in their shape and form because they also tend to be smoother without pockets because they're binding DNA or DNA complexes, and they're changing the expression of DNA. So very different than a lot of proteins that are just hitting receptors. And because of that, I think they've been ignored for a very long time, and they were always called undruggable. They're also complex, though. I think that should also be said, is that transcription factors, they allow for these complex programs of expression to change within the cell. And so those have been hard for people to understand. If you think about the way most academic labs look at a disease problem, they're usually highly focused on one protein or one pathway or one receptor that seems to be going wrong in the disease. and they'll focus on that one thing, and they'll look at what's upstream, what's downstream of that, and they'll make nice little pharmacology drawings that make everything look nice and simple. But the reality is most diseases, especially diseases of aging, complex diseases, there's a lot of things that are going wrong. Same thing with transcription factors, where there's just a lot of action downstream that happens because of them. And so I think that uh, those have been historically the problems. The nice thing and the fortunate thing, the reason we're working in this field at Junevity, is there's been a lot of technological advance that we think allows, for the first time, to really look at transcription factors as drug targets.
Well, what constitutes an indication that Junevity would pursue, and what are the specific characteristics they must have for you to think about going after them?
Yeah, so I think first and foremost, we look at it in a lens of we're focused on diseases that are driven by transcriptional dysregulation and diseases of aging. And so it really has to fit within that realm for us to say, want to be interested in it, feel like we can make an impact. Because the key at Junevity for us, the key technology breakthrough that we've made and we're building upon, is our Reset computational platform, which is a drug discovery platform. It allows us, for the first time, to look at these large scale transcriptional data sets in disease and look at regulon changes and figure out what will be a potential drug target, drug pathway that we can go after. So that's the high level lens that we're looking at for any disease indication. I think from a business biotech startup standpoint, because we are a small company, we think that next, where are the diseases or indications where we think we can make a big impact. So where are the underserved diseases? We typically focus on diseases that affect a lot of people. I think part of that's because transcriptional dysfunction diseases tend to be pretty widespread. It's something that happens to everybody's body, tends to show up in different ways, but it does lead to some of the biggest diseases that affect people. So where can we make a big impact on health? And then, from a business standpoint, where can we find a pathway as a biotech company to actually develop a therapy that we can then show works for a reasonable amount of time and money. And there's a pathway with the FDA to actually eventually help patients.
It would seem that cancer would fit right in there, but you don't appear to be pursuing cancers. Is that correct?
So we are certainly interested in cancer, and you're right. Cancer does fit within that criteria, absolutely. I think that our initial indications of type 2 diabetes and obesity, one of the reasons we were interested in those was they fit within the criteria we were looking at from a transcriptional change perspective, but they're also indications where you can do non-clinical studies and show pretty quick proof of concept. They're also very big indications where there's a lot of interest right now. Cancer, certainly there's interest in cancer. It's still a very big problem. There's tons of companies going after it. For us, that is something that we're looking at on the discovery side right now, and we intend to start pursuing preclinical studies in 2026. But it's certainly higher on our list, and it certainly fits within that rubric that we're looking at.
Well, let's talk about your platform technology. This is Reset, which is an AI-based system. This is based on work done by Janine Sengstack at UCSF to identify overactive pathways associated with aging and disease. She's a co-founder and chief scientific officer of the company. What's the input for the platform and what's the output?
Yeah, so Janine was the original, I would say, the original founder of Junevity. We have 3 co-founders, including myself here, but certainly Junevity is based on the work that she did with her thesis advisor, Hao Li at UCSF. And this was right around the time when the Yamanaka factors were discovered. Uh, uh, Dr. Yamanaka's paper came out showing that you could basically take any cell in the body and take it back to a pluripotent stem cell, which by altering the expression of 4 transcription factors. And that was not only led to a Nobel Prize, but it opened people's eyes up to the fact that transcriptional regulation is hugely important and can even guide the identity of a cell. And so at that time, Janine and Dr. Lee started thinking about how could you identify, how could you actually take all this transcriptional data, which is very large and complex, and start to evaluate it in a way that you can start to tease out what are the key transcription pathways that are going to be causing disease or associated with age. And so they did a lot of work looking at large scale data sets and figuring out how to analyze those. And one of the big things that the Reset platform does is it can take input from these data sets, which I should also mention are usually very rich. Because instead of just looking at one protein to see if it's mutated or not, you're looking at hundreds or thousands of proteins and seeing how they interact with each other. So it's really looking at the network effect. And that is the input where you have uh, millions or billions of data points coming from multiple subjects in a single data set. And then looking at the network changes in what we'll call the regulon. Regulon is the set of proteins, which could be tens or hundreds or even thousands of proteins that are being altered by the transcription factor itself. And then having that network point back to what is the transcription factor upstream that's actually driving all of this change consistently throughout the patients. And so creating this uh, looking for the pattern that is the real driver of the disease instead of just looking at a single protein and saying that must be the thing driving disease. It's, well, what's actually causing that protein to change, and all of the things around it that seem to be implicated.
Are you using public data sets for this, or are you generating the data from tissue samples from people with given diseases?
We're using public data sets. I think the really amazing thing about the Reset platform and about being an innovator in this space looking for transcription factor targets is that there is a wealth of public data sets out there, and the Reset platform and the real value that it holds is being able to use that existing data in a new way, to look at it from a network perspective and find essentially novel targets. I think that we're able to do this now because we're just starting off to using this type of machine learning approach for these data sets, and we're able to look at it for the first time and kind of get that low hanging fruit of targets for these big diseases where there are big public data sets. If we're successful, if our ideas turn out to be effective in treating disease, we start to generate therapeutic candidates that show real promise. I think there will be a point at which we grow as a company. We start generating novel data sets from tissue samples, because those public data sets we will have combed over. And at this point, we've already gone through 10 plus disease indications, and we've already started discovery on public data sets in those disease indications. So you can imagine in 5 years, we're going to have run through most of the things that do exist. But for now, it's actually a very ripe field to use the public data sets and get cool targets.
And how do you go from the output of the platform to a therapeutic candidate?
So the reset platform gives us usually a ranked list of potential transcription factors to target. And so then our job, this is what the other side of Junevity, outside of the computational side, we look at those targets and we go through a very rigorous and robust development development pathway, which is more similar to a traditional pharmaceutical approach. In that, we'll start off in cells, so doing in vitro experiments on usually the cell type associated with disease or the cell type that we're going to be targeting with our siRNA. And we do in vitro work to look at things like knockdown, things like transcriptional change within the cell. There might be disease biomarkers you can look at within the cell for a specific disease. And then we go into in vivo studies. We look for disease models in preclinical species. We also do a pretty very thorough effort on siRNA optimization. So, you have a certain gene that you're trying to target. It has a certain mRNA pattern that you're going to try to target. And we do a computational approach there. We usually get back about 2,000 different siRNA sequences that could target that gene, and then go through a robust, totally separate pathway to try to optimize down to a top 2 or 3 candidates, and then move those into potentially higher species if there's a good disease model that exists, and then end up with what we would call a development candidate or therapeutic candidate that would be ready to push into the clinic.
And have you identified a lead program, and what's the path to the clinic for you?
Yes, at Junevity, we have 2 lead programs. They're pretty much neck and neck in the development path so far, and one is for type 2 diabetes. So it's an siRNA we call it June-01, and it's an siRNA targeting the liver. So using GalNAc technology, which is very tried and true. There's a number of approved products using this technology. It specifically takes the siRNA from an injection into your belly, similar to how you would deliver a GLP-1, like Ozempic. But do a simple injection, goes into your liver, and then targets a transcription factor that we've seen preclinically leads to huge benefits in insulin sensitivity and glucose reduction. One of the keys for that program is that, based on the data we have thus far, in humans, it looks like it could be delivered every 6 to 12 months, which would be a big advantage for patient compliance and disease treatment overall. The second program is for obesity, and this one is also delivered via subcutaneous injection. It's an siRNA. It does target liver as well as adipose and muscle. And so far, preclinically, we've seen really, really exciting differentiated results with that. Again, we think it could be a low frequency of administration every 6 to 12 months, but it's that one's different than what the GLP-1s or things like Ozempic, Zepbound, in that instead of mainly relying on a satiety signal, it actually looks like it's doing more of a metabolic reset, taking your metabolism back to a healthier place, a true reset point, so that what we've seen preclinically, even if you come off of the medication, the effect still lasts, and so you're not seeing the rebound. And people could take this and actually have a healthier metabolism to allow them to keep weight off long term.
These are two large but well-served markets. What problems do you see with existing therapies?
So with type 2 diabetes and obesity, they both have had, with the GLP-1s, which have been approved in the past 10 years, uh, huge impact on disease. And those products, I think, are true miracle drugs. They're amazing. However, there are still issues with type 2 diabetes and obesity where additional medications we think could serve the market. And it's not just us at Junevity. I think you see this with large pharmaceutical companies are still saying right now, like, let's look at the next generation of therapies and how they can help with these diseases. I think yesterday there was a multi-billion-dollar acquisition of a phase 2 uh, clinical company who has a number of these next generation products in their
This is
pipeline.
the Pfizer deal you're talking about?
Yeah, Pfizer buying Metsera, and so I think there is this view that GLP-1s are amazing, but still problems, and still opportunity to serve patients. So type 2 diabetes specifically, um, there's still an issue with patients being on kind of a polypharmacy and a lot of things that reduce glucose, but not a lot of things that actually improve insulin sensitivity. So on the one hand, you want to reduce your glucose levels, and you can use things like metformin, which reduces gluconeogenesis. You can use SGLT2s, which basically make you pee the glucose out. Um, GLP-1s do a good job of diabetes and obesity, just preventing that preventing that progression. But once you are a type 2 diabetic, GLP-1s don't work as much. And I think the big issue that we see is patients just don't take their drugs a lot of the time. There's about 50% of patients, almost half of patients, have an issue with compliance on their medications. And so if you're not adhering to your medication, um, then you're going to have these high levels of glucose because it's basically like you take your meds, glucose goes down, you're off your meds, glucose goes up. And that it slows down the progression of disease, but it still is leading to these downstream effects, which, you know, downstream effects of diabetes are still hugely uh, impactful on the patient and on the healthcare system in general. And so we think that something you could take every 6 to 12 months could improve your insulin sensitivity, the way that your body is actually utilizing insulin, but also then lowering your glucose levels. It would help with that problem of patient compliance, and it would help people from truly progressing into the disease and not only save a lot of pain in the patients and downstream consequences, but it would also save the healthcare system at large a huge amount of money because those downstream consequences like chronic kidney disease, like diabetic blindness, like amputations, these are things that are very, very expensive. And that's where you end up, having society pay for the burden of the disease. With obesity, GLP-1s are amazing. Again, one of the big issues is that they have to be taken as a weekly injection. So adherence, again, is not great. I think in a study in the UK that was done, they said after 1 year it was like 64.5% adherence. After 2 years, it was about 60% adherence, with partial or full discontinuations of 50 to 60% after year 2. So you have people taking these, and it's great, and then they feel healthy, and then they stop taking them, and then the weight bounce back from GLP-1s is pretty intense. Most people gain back 70% of the weight within a year after stopping. And so with obesity, we think again, if you have an siRNA that can stay in the body, patient doesn't have to think about it. You inject once every 12 months, and you can keep that weight off for the long term because obesity and diabetes, those are decade long diseases. That's where the consequence happens. We think that that could really take GLP-1s to the next level. Our medications at Junevity, fully compatible and additive effects we've seen with GLP-1s. We think that it could help these medications over the long haul actually keep people in compliance and keep people away from the severe end of the disease.
You also have an interest in frailty or sarcopenia. Muscle loss is a big concern among people using GLP-1 therapies. Are you looking at that as an additive therapy?
Yeah, so one of the gene targets we are looking at seems to have a nice effect of weight loss, but more lean muscle preservation. And there's other therapies out there that look really interesting in this space too, like the amylin agonists, which Metsera, that they have an asset there that's very exciting. Um, but from an siRNA perspective, we think that improving weight loss to be a healthier weight loss, where muscle loss is lessened, or you can keep that strength. It's not only going to help people stay on the therapies longer because they won't feel frail, but when you think about the usage of these medications, there's huge benefit that can be had in people that are over 65. And if frailty and muscle loss, which you do see with GLP-1s, if that is part of um, the therapy, that all of a sudden becomes a bigger indication in the older population. And so I think, again, I don't see this as there's going to be one drug to rule everything. I think obesity specifically is a field that is just starting to get acknowledgement as a downstream driver of so many healthcare problems, including things like cancer and dementia, which you've seen risk reduction with the GLP-1s. Um, I think that as combination with the GLP-1, something like our siRNA Juno 2 could do a big job in improving frailty and really opening these types of therapies up for long-term use in an elderly population.
It's been a tough environment for startups. The company raised 10 million earlier this year. What was the discussion like with investors?
Yeah, it has been a tough environment for startups, and I think that that's just an unfortunate effect of the broader macroeconomic environment. So the stock market's been rough for biotechs. Uh, the that leads for, you have VC groups that have companies without a real exit opportunity or liquidity, and then they don't have money for new startups, and so as you said, Series A's have been really tough. I think the bar has just moved forward on everything, so that money is just not as easy. Um, Not necessarily a bad thing for the field overall. It's probably good to have those types of things every once in a while, and I think it will allow for truly strong companies to figure out ways to move through. I think for us, the talking with traditional biotech VCs, they essentially didn't historically, let's say 6 years ago, you could probably go with a preclinical idea that was really cool and get a Series A. I just don't think that's possible now. And I think they're looking for things that are clinic ready or in the clinic even. And so what we did as a company was we started talking to non-traditional VC, non-traditional investors, and you're not your biotech VCs. We started talking to family offices, which family offices there's a lot of them. They've started to become more interested in healthcare, biotech. There's a lot of traditional tech investors that have started to become more interested in uh, longevity, biotech, healthcare, and so we were actually. It was not incredibly painful to raise our $10 million that we announced earlier this year. And actually, we have raised additional money since then, based on the strength of our preclinical data. And we haven't announced that yet, so I'm not going to announce the amount yet. But it definitely, I think, having good data and being in an area with significant pharma interest and investment, it's still possible to raise money from VCs or non-VC investors to build a life science company. I think it's just a different route than what has historically been there.
Well, how far will existing cash take you, and do you expect to need to do another fundraising round to begin clinical studies?
So right now at Junevity, we have enough money. We have a runway takes us through 2027, which is for a biotech company, that's a decent amount of runway and capital, and that does include an initial clinical study in a lead program. So far, we feel like we will be able to take our lead assets, take at least one lead asset, and get it through human proof of concept in initial clinical work. And that's what we feel. We might raise money before that or on the way if we can get investors excited. We might be out fundraising again, in the next year. But we feel like we have a plan, we have some exciting assets, and we should be able to get through human proof of concept with the capital raise so far.
John Hoekman, co-founder and CEO of Junevity. John, thanks so much for your time today.
Yeah, thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.